So I will start my class today, and I will be talking about Mendel's laws and some extension on Mendelism. So with this, let me present my screen. Is the screen visible?
Yes, sir.
Okay. So as you people may have briefly learnt about what Mendel's law is. So basically, Mendel is considered to be the father of genetics. In around the year of 1865 to 1865 to 1869, around this time, he did certain experiments with pea plants. He did some crosses, and he got some results, and the results were analyzed statistically. And the results were analyzed statistically. So basically, it was basically what he did was he did certain thing known as a technique known as cross pollination. And on the basis of these experiments, he gave three laws. The first is known as the principle of dominance. The second is known as the principle of segregation. And the third is known as the principle of independent assortment. Now these work was performed by Mendel in around this time, and then Mendel, he expired in the year 1884. He died. He had some liver problems in Brunn. He was an abbot, father, priest over there in the Brunn monastery, and over there he used to conduct his experiments, and so he died, and the local newspaper reported that their beloved father Abbot Mendel had died, and he was particularly known for breeding one of the best varieties of fuchsia flowers. So his brilliant work with pea plants, it was not mentioned also very much. So Mendel was lost for 2.5 decades. He was then rediscovered around 1901, 1903 around. There were three scientists who rediscovered Mendel. Rediscovered Mendel. Among them, it's not important you know their name. One of them you can know. One of these scientists was the famous De Vries. So these people, and there were another two, Shamark, and there was one more of these scientists who independently rediscovered Mendel. So they did this experiment, they did their experiment, and then they suddenly found that in 1869 Mendel had already performed all these work. And so they tried to reduce his stature, and said that they did the work mostly and all these things, but it did not work. So approximately 25th year after Mendel's death, the square in Brunn was named after Mendel, and Mendel was re-christianed into, considered to be called the father of genetics. Many people criticized him. So these are the three laws which he gave, crossing pea plants. During Mendel's time, you have to remember one thing. Mendel used the term factors, since the concept of gene was not given then. It was introduced, gene was introduced by Johannesson in 1905. The term 1909, he gave the Johannesson gave the term gene. Gave the term gene. Mendel used the term factors. So after keeping this in mind, let us go to the next part. And now we will talk about principle of. Now what is this principle of dominance? Now before we consider, let us consider the term what allele is. Allele is defined as contrasting forms of the same gene in same loci of the chromosomes. Now I am talking in terms of modern concept, using the chromosome theory of inheritance. So this is, say, these are the two homologous chromosomes. This is from the mother and this is from the father. And let's say this is the same region. This is, say, chromosome 21. Let us say this is chromosome 21. They are in the same region, this chromosome 21, the same place. And say they control one thing, and that is known as height of the plant. One may be wild type and one may be dwarf. So the plant can be tall or the plant can be dwarf. There can be these two contrasting forms. So you can find these two contrasting forms over here. So and say if on both the chromosomes it is capital D, this is a homozygous tall. This is also homozygous dwarf, and if it is, this is a heterozygous. As per this law of the principle of dominance, when two pure forms, say capital D tall, it is crossed with a pure form dwarf, the first filial generation which you get, this heterozygous, they are all tall. So this D is dominating over short. This is the principle of dominance. Next we go to the principle of segregation. So this cross was done. Capital D into small d, F1, all was tall, and now they were again crossed among themselves. What they got was eventually in the F2, this was tall, and very important, they got this dwarf back. This dwarf again re-emerged. Dwarf character which was lost in the F1 re-emerged in F2. So the principle of segregation was, he suggested that therefore DD into, when this particular cross was done, the gametes just segregated. Sorry. They separated. So when this particular this came out and then this separated, this form re-emerged. So this is the principle of segregation, that the gametes, they separate or segregate. And this type of a cross using one character is called a monohybrid cross. This type of a cross is called as a monohybrid cross. Now Mendel did a dihybrid cross, where he used two different characters. First was shape of the pod, and the other was the color of the pod. So he had a yellow, round, and green, wrinkled. He had these two types of pods and he crossed them. So what was the result of this cross? Yellow round, capital G, that is yellow, and round is this was yellow round, homozygous yellow round. And this was crossed with, sorry, this is capital W. Let me erase it. Capital W, capital W. This was crossed with green, wrinkled. Small w, this was now when this the gametes they segregated in the F1 generation, they segregated and they formed GW, DW. So basically this was heterozygous yellow round in the F1 generation. And now when they were selfed, they were crossed among themselves. What they found was now I am using a different color. In the F2 generation, you had the independent segregation taking place. Gametes separated. G GW, capital G, small w. Then you had small g, capital W. And now you draw the box. Now what you find after you draw these box, when the crossing took place, this was GGWW. This was yellow round. Before I draw all these, let me first write down everything. So what are the combinations which you get? Here you get GGW small w. You get GG capital W W. Then you have here GG capital W small w. Then you write here small w small w. Then you have GG small w small w. So I'm just placing them, writing them one by one. GG capital W small w. Again small g capital W capital W. Then small g capital W small w. Then you have capital G small g capital W small w. Capital G small w small w. Small g capital W small w. And finally you have small g small w small w. And now you find what is the pot shape. This is yellow and round. This is also 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 yellow and round. For the first time this is yellow and small w. This is yellow and wrinkled. So for the first time a new assortment comes out. Yellow wrinkled. It was not existing. New assortment comes out. Assortment means new combination. So this is the independent assortment, and this happens independently. One is not controlling. So the presence of it being yellow and round, yellow is not controlling the character being round, or green is not controlling the character being wrinkled. It is independent. Yellow and wrinkled can exist together, and for the first time you see yellow wrinkled coming together. This is again yellow round, and again you find this is yellow wrinkled, another wrinkled you get. Then you find yellow round. This is also yellow round. Now this becomes green. Green, and this will be green. A new character, green round. This is also a new assortment. Green was always wrinkled. It was never round. Again this is green round. This is yellow, again yellow round. This is again yellow wrinkled and green round, and finally green wrinkled. So what is the ratio of, let us have a look at the ratio of what happens. So let us see what is the ratio of how many yellow are there. 1, 2, 3, 4, 5, 6, 7, 8, 9. You have 9. Then how many yellow wrinkled are there? 1, 2, 3. How many green round are there? New combination, that is 3. And how many green wrinkled are there? 1. So the ratio comes in this dihybrid cross as 9 is to 3 is to 3 is to 1. Mendel observed this by counting the number of pods and all these things. So Mendel, this type of cross is known as dihybrid cross, and this proved the principle of independent assortment. And what this means, that presence of yellow color did not influence the pod into being round. It can be wrinkled also. At the same time, presence of green color did not influence the pod into being wrinkled. It can be round also. So this is Mendel's laws. Now let us go to the next part, which is extension of Mendelisms. Now we will talk about so as we know, definition of allele, you can write it as an allele is one of a number of alternative forms of the same trait or gene. Now we are coming to the modern term. It is present in the same genetic locus. Gene, genetic, chromosomal locus. So, for example, eye color in Drosophila. It can be red or white. You can write it as small y, but they are located in the same place. Eye color gene. The gene is for eye color. And red and white are alleles. Now we know that, say, আমার কথা শোনা যাচ্ছে?
হ্যাঁ স্যার।
ঠিক আছে। Red eye color. Why is the red eye color? This is because the gene for red eye color, it codes for mRNA, which codes for a protein which is responsible for red eye color. It makes a pigment which is red eye color, gives red. So if there is a change in the DNA sequence, it can very well be red eye color is not made. Absence of red, it becomes white. So, however, red and white are alleles. They are different forms of the same genes, eye color, and the same genetic locus. Now, few things which we must see what happened in snapdragon. It's a flower, a phenomenon known as incomplete dominance takes place. What happened over here, we are talking about the flower color. So this is the phenotype of the flower, genotype, and amount of gene product. We talk about this different. What happened was, when red and white, they were crossed, red flower and white flower, they crossed, you expected that you would get red flowers because red is dominant, but instead what you got was, you got a pink flower. So say red is capital W, white is small w, and pink is so the amount of gene product, so you have two of this, so the amount of gene product is 2 times 2x, this is 1x, and there is no red colored gene product. So it is not completely just a mixture. The red is not completely dominant over white. Thus pink color is formed. This phenomenon is known as incomplete dominance. Now let us look at the case of what is codominance. We have a blood group known as blood group L, blood group M. We have, apart from ABO, we have this blood group M and blood group L and blood group M. So if you look at the genotype, LM, both of the father from mother, LM. So the blood type or the antigen which is present, blood type is M. Now if the genotype is LN, blood type is N. But if it is LM, LN, the blood type is both MN. Both of them are present. And this phenomenon is known as codominance. This codominance also you can find it in blood between blood group A and B, where A is dominant over O, B is dominant over O, and A and B are codominant. So people with A and B, mother and father, they become AB blood group. This is the concept of codominance. Along with this, we come to the concept of multiple alleles. Now we were dealing with forms which only two forms we were dealing. Of a particular trait, red, white, this thing. Now let us we are talking if there are two or more forms, A, B, O. These is multiple alleles. This ABO blood group, you have antigen for A, IB, and you have IO. You have these three group present, three types of forms present. In this case, A equal to B, A is dominant over B, A is dominant over O. This is multiple so A is dominant over O. So this is multiple allele. Next we come to a phenomenon, the last part we come to a phenomenon which is very important for your examination. Although in my note you will find other parts also, you should read briefly for your competitive exams and stuff like that. That is nothing much, few more details and examples. We are talking about known as epistasis. So what is epistasis? Here, when two or more genes influence a trait. So one phenotype, this is one phenotype. You have two genes, one A and one B. They are affecting, both of them are needed to control the particular phenotype. When two or more genes influence a trait, an allele of one of the gene has an overriding effect. Say this can be AA, BB. It could be very well that B is dominating over all the proceedings of all the small b. One allele, it has got an overriding effect. So this B in this case is said to be epistatic to the other gene. So classic example is you can find it in the sweet pea, Lathyrus odoratus. Let's see what happens over there. Sweet pea, Lathyrus odoratus. Here the flowers, they can be either white or they can be purple. So when it is purple, then a pigment known as anthocyanin is present. Anthocyanin gives a purple color. And if not present, then flower is white. Simple. So Bateson and Punnett, they crossed two different white varieties with white flowers. Two varieties of white flower. So white flower, one was crossed with light flower 2. Surprisingly what came was the F1 hybrid, they had all purple flowers. Shadaye shadaye lal holo. So this is very strange. It's not supposed to happen like this. White should give rise to white only. Can be possible? How is it possible? So when these hybrids were intercrossed, so these purple flowers they were intercrossed, you expected a 9:3:3:1 ratio, but this was not found. Rather what you found was a 9 purple is to 7 white ratio. So they explained the phenomenon they proposed two independent assorting allele C and P are involved in anthocyanin synthesis. Now I am highlighting this part. So C had a small c and capital P had a small p. So they had a recessive allele. So when it is CC or PP, or when it is C, This removes pigmentation. Or same here. So if you give this phenotype, if I give this hypothesis that they had this recessive allele like this, so the parental variety, white flower parental variety, small c, capital P was crossed with capital C, small p. So both C and P, they are needed for purple flowers. So this flower will be a variety of white, this will be also a variety of white, and C and P are independently assorting. Assorting like your yellow and round and green and wrinkled, they were independently assorting, they are also independently assorting. So when they were crossed, they produced CC PP double heterozygote. They produced PP double heterozygote. So in this system, a dominant allele from each gene is necessary for synthesis of anthocyanin. So both, at least you needed one capital C and one capital P. At least this is needed. Needed for purple flower / anthocyanin synthesis. So when you do this in the F2, 9 out of 16, they are either capital C, it can be a capital C or small c, capital P, or it can be anything over here, either this means they are white, purple, and the remaining 7 out of 16 they are homozygous for at least one of the recessive alleles. So let us draw this cross once more. In the parental generation, P, white, capital C, small p, crossed with also another variety of white, small c, capital P. Result F1 purple, capital C, capital P, small p. Now if we draw the box, let's saw the gametes. They are self now. Capital CP, C small p, small c capital P, small c small p. Similarly over here I draw the same thing. Small c small p. Then I draw this box again. Now let us draw this dihybrid cross again. Capital C, capital P, capital P. Then I will mark the colors then. Capital C, capital C, capital P, small p. Capital C, small c, capital P, capital P. Capital C, small c, capital P, small p. Capital C, capital C, capital P, small p. Then we have CC, capital C, small p, small p. Capital C, small, capital C, small c, capital P, small p. Capital C, small c, small p, small p. Capital C, small c, both are capital P's. Then you have capital C, small c, capital P, capital P here. Very good. Then you have small c, capital P, capital P. You have small c, capital P, small p. Then you have capital C, P, P, small p, small p. Capital C, small c, and small p, small p. Then you have small c, capital P, small p. Small c, small p, small p. Now let us see which are purple. I am drawing it with pink. This is purple, 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 purple. This is also purple, this is purple, this is also purple. This is also purple. Are purple, this is purple. So 1, 2, 3, 4, 5, 6, 7, 8, 9. So 9 are purple and the rest are white. And this phenomenon is known as epistasis. The next thing, the last thing which we will talk about is pleiotropy. কোথায় গেল? Pleiotropy. Now what does pleiotropy means? In Greek it means to take many turns. A phenotype can be influenced by many genes. Many genes. Or a gene can be influenced by many phenotypes. So, when a gene affects many aspects of the phenotype, it is said to be pleiotropic. For example, the gene for phenylketonuria, PKU. So, what is it? The primary effect is, what happens is, it's when it is a recessive mutation, দুটোই এক থাকে, recessive homozygous mutation, একটা a toxic substance is formed, and this brain impairment গন্ডগোল হয় মাথায়, brain development এ. Now, this same problem, this PKU, this also interferes with melanin synthesis. Melanin synthesis, color of the hair. So, the same gene, one gene two phenotypes. This is pleiotropy. This is one aspect. আর কিছু আছে? Okay, last one, but not the least. We are talking about influence of the environment. In Drosophila, there is a mutation known as Shibire. What is the Shibire mutation? So, flies which have got this Shibire mutation at 25 degrees centigrade, these flies are viable and fertile. They are normal, they behave normal, they are normal. These are extremely sensitive to sudden shock. So when you shake the flask, these flies become paralyzed and fall temporarily. Paralysis হয়ে ভ্রাম করে পড়ে গেল, অন্যরা আবার উড়ে চলে গেল. These flies, they become paralyzed and fall. So the environment is a shock environment. Now you increase the temperature to 9 degrees centigrade. All these flies fall to the bottom and die. With this I actually end my class today. Now just asking if you people have got any questions. Kono questions ache? Any questions?
No, sir.
Okay then.
না স্যার।
Okay, then I end my class over here. I will tell you about my next class.
ওকে স্যার। স্যার এইগুলোর আপনি যে নোটগুলো কালকে পাঠিয়েছেন ওগুলো
আজ
করলেই হবে
ভোরবেলা
তো?
পাঠিয়ে দিয়েছে। হ্যাঁ যথেষ্ট।
হ্যাঁ।
যথেষ্ট।
ওকে স্যার।